Today, we're diving into something that honestly sounds like it's pulled straight from a sci-fi movie. We're talking about programming living bacteria to become microscopic cancer assassins. These are tiny biological machines that can actually hunt down and destroy tumors, all while leaving healthy cells completely untouched. So, let's get into how this incredible technology actually works.
We have to start with this fundamental question, one that's haunted oncology for decades. Chemotherapy is powerful, for sure, but it has always been a blunt instrument. So what if we could finally make it smart? What if we could give it a targeting system so precise it could tell the difference between a cancer cell and the healthy one right next door?
And that really is the core problem with traditional chemo, the collateral damage. It's a systemic treatment, right? Which means it floods your entire body to kill off those fast-growing cancer cells. But the thing is, lots of our healthy cells, in our hair, our stomach, our immune system, they're also fast-growing. And the result? Just awful toxicity and those debilitating side effects we've all heard about.
Now, scientists have been trying to solve this for a while with something called a prodrug. Think of it like a weapon with the safety on. It's an inactive, totally harmless version of a chemo agent. It only becomes deadly after it's metabolized inside the body. It's a great idea, a definite step in the right direction, but that huge challenge still remains. How do you make sure it only activates right at the tumor and nowhere else?
Okay, this is where the story gets really fascinating. The answer might not come from some new synthetic chemical cooked up in a lab, but from a living organism, a natural ally that's, well, it's already a part of our daily lives and even living inside our own bodies.
So meet our hero, Lactobacillus plantarum. And yeah, you've definitely run into this before. It's a common probiotic, the good bacteria you find in things like yogurt. But this little guy has a hidden superpower. Researchers discovered it has this natural built-in attraction to certain types of cancer cells.
And let's be clear, this isn't just some random chance. This bacteria has what you could call a homing beacon that lets it navigate through the body and specifically seek out tumors. And this incredible ability all comes down to a lock and key system at the molecular level.
Okay, I know this slide looks a little complex, but the idea is actually pretty simple. You see, many cancer cells, especially the nasopharyngeal carcinoma they studied, are coated in a specific sugar called heparan sulfate. Think of that as the lock. Well, our bacteria, Lactobacillus plantarum, has a protein on its surface called OppA, and it acts as the perfect key. It physically binds to the cancer cell's surface, letting it stick right onto the tumor while just floating past healthy cells. This is the first crucial layer of its smart targeting system.
So, we've got our living drone, our delivery truck that can find the target all on its own. But a truck is pretty useless without any cargo. The next obvious step was to arm it, to give this bacteria a weapon it could deploy once it reached its destination.
So how in the world did they arm a bacterium? Well, it's this brilliant three-step process. First, they added a kind of molecular superglue called streptavidin all over the bacteria's surface. Second, they took a really powerful chemo drug, SN38, and made an inactive prodrug version of it with a little handle made of biotin. And finally, they just mixed them together. The biotin handle on the drug snaps right into the streptavidin superglue, and just like that, you've given the bacteria a little chemotherapy backpack.
In this schematic right here, this is the final product in all its technical glory. What you're looking at is our Lactobacillus plantarum, now fully armed. It's covered in that streptavidin glue, and attached to it is the inactive prodrug, called TLSN. It's literally a living weapon, ready and waiting for its mission.
But here's the second, and you could argue the most important layer of safety in this whole system. Even after the bacteria finds the tumor and latches on, the chemotherapy is still in its harmless, inactive prodrug state. It can't hurt anything. It's like the Trojan horse has already breached the city walls, but the soldiers inside are just quietly waiting for the signal to attack.
And that signal, it comes from the tumor itself. See, tumors create their own toxic little ecosystem around them, called the tumor microenvironment, or TME. This area is chemically completely different from the rest of the body. It's chaotic, it's acidic, and it's absolutely flooded with high levels of oxidative stress.
The key component of that oxidative stress is a group of molecules called Reactive Oxygen Species, or ROS for short. Healthy parts of your body have very low levels of ROS, but the tumor microenvironment is saturated with them. And here's the genius part. The scientists designed the prodrug safety lock to be picked specifically by ROS and nothing else.
So this slide gets into the chemistry, but here's the aha moment.
When our armed bacteria gets to the tumor, that high concentration of ROS acts like a chemical trigger. It literally snips the chemical bond holding the prodrug together, releasing the now fully active chemotherapy SN38. The tumor itself triggers its own destruction. It unleashes the weapon precisely where it's needed most, and only where it's needed most.
So, the big question, does it actually work? Well, in a mouse model with nasopharyngeal carcinoma, the results were just stunning. This bacterial therapy led to a 67% inhibition in tumor growth compared to the control group.
And to put that into perspective, this represented a 54% increase in the drug's effectiveness compared to just injecting the same dose of the active drug, SN38, on its own. So the delivery system didn't just work, it made the drug dramatically more potent.
Let's just look at this side by side, because it really highlights the massive shift here. Conventional chemo, high systemic toxicity. This bacterial therapy, it's inactive throughout the body. Old chemo damages healthy organs, like the liver and kidneys. This new method is activated only by the tumor. That means way less toxicity and a much, much better safety profile.
And the efficiency is it's off the charts. The researchers noted that because the delivery is so incredibly precise, the amount of chemo agent they needed to get a significant result is 750 to 2,000 times lower than what's normally used in these kinds of studies. That is a massive reduction in the drug burden on the body.
And the potential here is just enormous. While this study focused on nasopharyngeal carcinoma, that lock, the heparan sulfate marker, is also found on other types of cancer cells, including some bladder, lung, and even gastric cancers. This opens the door for this technology to be used in the fight against a whole range of deadly cancers.
What we're really seeing here is more than just a new drug. It's the proof of concept for an entirely new class of therapy, living medicine. We are moving from carpet bombing our bodies with chemicals to deploying programmable biological allies that can execute complex missions with just unbelievable precision.
It really leaves us with this profound question. For so long, we've thought of bacteria as the enemy, something to be destroyed. But what if the trillions of microbes living inside us right now aren't just passengers? What if they could be trained to become our most powerful guardians? The future of oncology might be less about chemistry and a whole lot more about biology.